Hi, my name is Arthur Thaon. I am a postdoc researcher at Queen's University Belfast. Today on this video I will give a brief talk about my poster, Vaccine Sequence Design, De-risking Autoimmunity with Preservation of Function, Stability and Efficacy. One of the major issues in vaccine design is when the antigen protein mimicries human protein. It may lead to a confusion of the immune system and by consequence a rise of the autoimmune risk. Here we developed a process to de-risk a protein sequence and verify its stability after mutations. The process is divided into two parallel phases. The first one focuses on the search of human similarity and reduces it, while the stabilization phase on the construction of the 3D structure from the inquiry sequence and a template. Its stability is checked during the molecular dynamics simulation, and we also include a retro feedback loop to optimize the inquiry sequence. Human sequence similarity is tracked using PsyBlast tool. The e-value indicates if the inquiry and the subject sequence are homogeneous or not. The percentage of identities indicates when amino acid residues shares similar physical-chemical properties at the same place in both sequences. The mutated amino acid residue is represented in red. We also developed a software to identify contiguous regions of identical sequence between a query protein sequence and the human proteome. 7-mers are unlikely to have high affinity to the MHC class I peptide binding pockets. A 7-mers matches to the reference proteome might contribute to 8-mers or 9-mer for some individuals due to the variation across the population. T cells recognize peptides presented at the cell surface by MHC proteins that have peptide binding cleft. They recognize peptide from 8 to 10 amino acids residue for the class 1 and 13 to 25 amino acids residue for the class 2. The screening of the T cell epitopes was made using TP tool and NetMHC pan. The ranking is based on predicted IC50 value. The percentile rank column identifies the peptide's predicted binding affinity. Lower values indicate stronger predicted binding. Introducing a mutation inside of the inquiry sequence may disrupt the protein. Also, some proteins have physical and chemical characteristics that tends to make it difficult to administrate in vivo. To identify the amino acid residue to mutate, the original and the mutated sequence are realigned on chart view using the cluster O program. A color scheme corresponding to a specific physical-chemical characteristic, for example here the hydrophilicity, is applied to all of the sequences. The reconstruction of the 3D structure of the mutated sequence was done by homology modeling. 5 structures were proposed per inquiry sequences by the program modeler, ranked by normalized dop score. The lowest dop score structure was taken as input for the molecular dynamics simulation using GROMACS. A force field Amber 03 was applied to the structure and the complex was neutralized by adding ions of sodium and chloride. After energy minimization, the complex was temperature and pressure equilibrated. Each molecular dynamics simulation was run for 50 nanoseconds. The trajectory, the root mean square deviation, the root mean square fluctuation, and the radius of gyration were recorded. With the RMS fluctuation and the protein-protein interactions analysis, provided by the plot, the region subject to the movement and instability are mapped using a gradient color. Van der Waal and hydrogen bondings and electrostatic interaction are represented in space thinning. In a similar way, the hydrophobic interactions between the two chains of the protein are represented by a code color, and the amino acids which are not involved in any kind of interactions are represented in gray. The predictions were sent to our industrial partner. The company was able to express a mutated protein which has a similar profile with the original protein in the UV visible spectrum. The yield of the mutated protein was higher than the original protein. In conclusion, the predicted mutated sequence was correctly expressed and folded with a better yield. In summary, we developed an innovative process to reduce the human similarity of the increased sequence. This process was divided in two phases, one for the derisking and the second for the stabilization. The process was tested using sequence provided by our industrial partner. The protein was expressed and its yield was increased. Thank you.